Hi class, we're moving on with infection. Here's our intro to infection. So infection involves microorganisms. These are small living forms of pathogens, forms, and they include bacteria, fungi, protozoa, viruses. Many can grow in artificial culture medium, so we can grow it in the lab and we can look at it. A lot are non-pathogenic, which means that they don't cause any disease unless conditions change. And so we have our own microflora, a normal flora on our body and in our digestive tract and places like that. And they're often beneficial and keep out the pathogenic microorganisms. The pathogenic microorganisms are the ones that cause disease. So pathogens cause disease, and they do because of their characteristics and how they divide. So there's a lot of different types of microorganisms. Here we see at the top, these are bacteria, and they're usually named for their shape. Coccus is circle, Bacillus is a rod, Vibrio and Sporella are curves. Pleomorphic means they have various different shapes. Spirochete makes a spiral. Diplo means two. So this is Diplococcus, is two cocci. Staphylo means clusters or clumps. And then strepto means a chain. And then palisades means it looks like a fence. And a tetrad means four. Down here we see here's a virus. And it is enveloped into a glycoprotein. It has an envelope. And then on the inside there is a protein core. And then there's some type of nucleic acid that carries the viral genes. Okay? And the glycoprotein helps it attach. Over here, here is a fungus, and we can see this is a Chlamydia spore. You can see the spore that grows out, and it's going to form these blastospores, and they will go out and invade. Okay. Bacteria are classified as prokaryotes, which means they do not have a nucleus. So that's no nuclear membrane, no nucleus. They do have DNA, and they are alive, and they are cells. They function metabolically, and they reproduce. They divide what's called binary fission or asexual reproduction, where they just double their DNA and then they divide in half. They have a very complex cell wall structure, and the cell wall protects the inner cell membrane in the cell. They do not require living tissues to survive. Even though there are a few bacteria that do live as parasites, they don't require it to survive most of the time. And they vary in size and shape, and we saw those shapes earlier. So here's the reproduction. In the vegetative state, it's just living, eating, breathing, and then it divides into proliferates by dividing in half, and we end up with two identical daughter cells, and those daughter cells can divide, and they can divide. Bacteria have a very quick time frame for division, and they can multiply pretty rapidly as long as the conditions are right. The major groups of bacteria include the bacilli, which are the rod-shaped organisms, the spirochetes that have spiral forms, the cocci, which are spherical, and they're called diplococci, which are two together, streptococci, which is a chain, and then staphylococci is a clump. The structure of a bacteria we can begin with the cell wall. It's rigid. It protects it and gives it its specific shape. There's two major chemical compositions that we discovered of bacteria, which is called gram-positive, gram-negative, and we discovered it by they stain a little bit different using a gram stain. So if they stain one way, they're called gram-positive. If they don't stain, they're called gram-negative with the gram stain. This is important because they do have different characteristics, and we can use different antimicrobial therapies depending on whether they're gram-positive or gram-negative. The cell membrane is located inside of the cell wall, and it is selectively permeable just like our cell membrane. But we don't have a cell wall, so this gives us a target to actually do antibacterial drugs, is because it can attach to, if it can only work on the cell wall, it will not bother us. So we can see here how the antimicrobial drugs work on the different sites of a bacteria. We can see one of the major ways is through is by disrupting the cell wall and causing defective synthesis of the cell wall. This is what penicillin does. It's a bactericidal, and it will interfere with the cell wall. We can also affect the metabolic pathways of the bacteria, and sulfamides do that. They affect the metabolites. And then we can also interfere with the chromosomal DNA and protein synthesis, and that's going to interfere with the process of making proteins. And tetracycline does this. That's called bacteriostatic because it slows down the production of the bacteria. Bactericidal means it kills it. And then we can also have cell membrane damage like polymyxum, which damages the cell membrane. All these are going to slow down, inhibit, or kill the bacteria. Here we see there's a capsule or slime coat, and we have the cell wall. There's a strand of DNA, there's a cytoplasm, there's a cell membrane. There may be fimbriae or pili that help it attach and adhere to other surfaces. And there may be a flagella or may not be a flagella. And there are ribosomes inside of the bacteria. So the external capsule or slime layer is found in some bacteria. It's outside the cell wall and it adds additional protection. The flagella helps it move or attach to the cell wall, is attached to the cell wall and maybe one or more, and provides motility. The pili or fimbriae are tiny hair-like structures that can assist with attachment to a tissue for invasion so it doesn't get slayed off, or it can also help transfer DNA from one bacteria to another so they can pass traits along. The cell membrane inside the bacterial cell wall is selected permeable, like we said. The cytoplasm contains one chromosome in a circle versus ours is a double helix, so there's only one long strand of DNA. There's ribosomes and RNA that help to carry on the functions of the bacteria. And there's also maybe some DNA fragments, and they're called plasmids. These are not part of the chromosome, and they're fragments, and they can exchange DNA with other bacteria through the process of conjugation. Some bacteria have toxins. Exotoxins are ones that are produced and secreted out from the bacteria. These are usually produced by gram-positive bacteria, so they can produce these exotoxins. And that's part of the thing that gives them their pathogenicity, is that they can disrupt the tissue and cause damage. Endotoxins are toxins that are found within the cell wall of gram-negative bacteria, and so they stay inside the cell wall until we kill it, and then they release it. So this is one of the things you have to watch out with gram-negative bacteria. If you kill a lot of gram-negative bacteria all at one time, it releases all these toxins into the system and can cause things like septic shock if it has a vasoactive compound. And then we also have enzymes, and shock is just another word for super low blood pressure. And then we have enzymes that the bacteria can produce that can damage tissue and spread infection. Bacteria can divide, and we saw them go from one bacteria to two. If the conditions change and it's not in the best environment, some bacteria can go into a dormant form or a latent form, and that's called a spore. And these spores usually have very little fluid inside of them and can exist for a long period of time outside of their normal happy environment. These spores are also highly resistant to heat and disinfectants. Therefore, they say they're more resistant. The next microorganism that we're going to talk about is the viruses. And viruses are small, obligate intracellular parasites. They're obligate, meaning that they cannot reproduce and live outside of their host. And they only reproduce using the machinery of the cells that they are infected. Viruses have a protein coat or capsid, and that coat can come in various shapes and sizes. It can usually mutate pretty rapidly. The virus also has has a nucleic acid, which can be DNA or RNA. And one of the ways we classify it is by whether it's DNA and RNA. And then some viruses are RNA-containing viruses that can actually take the RNA and turn it back into DNA. And it uses an enzyme called reverse transcriptase enzyme. And it turns the RNA back into DNA in the host organism. So here we can see a couple viruses. We can see the capsid and then the protein inside. And here's the protein coat here and the envelope. This looks more like a HIV virus and has the receptors that can bind to specific cells. Here's a few different viruses. So, viral infection. The virus attaches to the host cell, and the viral genetic material enters the cell. The DNA or RNA jacks the cell and takes over its machinery and control of the cell. The virus DNA or viral RNA uses the host cell to make the proteins and nucleic acids to build more viruses. The new viruses are then assembled, and then the cell bursts and releases, or the cell lyses and releases the new viral particles. That's one thing that can happen. Or it can produce the viruses and then they bud off and get released into the environment. So here's a case of the viruses budding off and being released. Okay. So there's the virus. It attaches to the host cell. It releases its DNA and it hijacks the system. The host cell reproduces the viral DNA, copies it, and makes the proteins that are necessary. And the host actually assembles the new viruses. And then they bud off and release the virus to new host cells. We can have infections, viral infections that are latent. Latent is when the virus enters the cell as an active infection, and then the viral proteins are produced and inserted into the membrane. And this may stimulate an immune response and destroy the cell. But if the virus does not, it's called a latent infection. Okay, the virus may reproduce actively if the immune system is depressed. And in a latent viral infection is dormant until it can go into an active infection. So it lays dormant, latent. Let's stop right here.